Welcome to the fertilization module. In this talk, we're going to learn about some aspects of internal fertilization. We will begin with looking at different types of fertilization and then focus on internal fertilization in the context of mammals. Depending on where fertilization occurs, fertilization can be external or internal. When fertilization occurs outside the body of the organism, it is called as external fertilization. In this case, the egg or the ovum is laid outside the organism's body into the environment. This is seen in a variety of organisms that include sea urchins, frogs, and fish. On the other hand, when fertilization occurs within the body of the organism, it is called as internal fertilization. In this case, the ovum or the egg is present within the body of the organism. Internal fertilization is observed in different organisms like mammals, reptiles, and birds. In this talk, we're going to focus on internal fertilization in the context of mammals. Now, in the case of mammals, the female gamete development occurs in the reproductive structure called as the ovary, and that is the part that is shown in the blue box. So the figure shows parts of the female reproductive system, and in the ovary is where we actually have the oocytes undergoing development. Now, fertilization in mammals occurs in a structure that's called as the fallopian tube, that is shown in the blue box. Within the fallopian tube, there is a region called as the ampulla, which is shown in the small blue box. And this is where fertilization occurs in mammals. Now, the developing oocyte that is made in the ovary has to move from the ovary to the oviduct to meet the sperm. Thus, in the case of mammals, fertilization is internal, unlike sea urchins where they showed external fertilization. Since fertilization is internal in mammals, we need to have the translocation of the oocyte and the sperm into the oviduct. Now, the oocyte is released from the ovary during ovulation, and it doesn't come alone. It is actually surrounded by the cumulus cells. The cumulus cells are required for the movement and binding of the oocyte to the oviduct. Now, ciliary beating as well as muscle contractions aid in the transport of the oocyte complex to the ampulla that is present in the oviduct. Now, this is how the oocyte is able to go to the proper place. In the case of the sperm have to translocate from the vagina to the oviduct, and they're able to do so through flagellar action. So sperm are motile due to the presence of flagella, and hence they're able to use that flagellar action to move towards the oviduct. There are also uterine muscle contractions that occur that help pump the sperm into the oviduct. In fact, it has been shown that if you don't have uterine muscle contractions, then the sperm take a much longer time to reach the oviduct. The sperm also show rheotaxis, which is the migration against a liquid flow. The liquid normally flows from the oviduct to the uterus, and the sperm are able to swim against this liquid flow to go from the uterus to the oviduct. This process requires or uses the Caspar calcium channels that are present in the sperm to sense the direction of the current. Now, the sperm need to complete their development before they undergo fertilization. So sperms, even though they undergo majority of the development in their testes, they do not complete their development there. They complete their development in the oviduct, which is present in the female body. These final stages of sperm maturation is called as capacitation. The sperm binds to the oviduct to become capacitated. And once it becomes capacitated, it is then released and then it can enter the ampulla. The binding of the sperm to the oviduct helps in extending the lifespan of the sperm. It also prevents too many sperm reaching the egg at the same time, and hence can be considered as an indirect way of preventing polyspermy. The capacitation of sperm is required for the sperm to become competent to fertilize the egg, and hence if capacitation does not occur, the sperm is unable to fertilize the egg. There are a few changes that happen in the sperm during the process of capacitation, and one of them is the changes in the lipid composition. So during capacitation, cholesterol is removed from the plasma membrane of the sperms, and this changes the lipid composition. And this is what is shown in the blue box in the figure, where you have proteins that are able to displace cholesterol from the plasma membrane of the sperm. There are also changes in the cell surface proteins and carbohydrates expression, and many of them are lost. And it is thought that this loss of cell surface proteins and carbohydrates help in the unmasking of different recognition sites that are present on the sperm. There are also changes in the membrane potential of the sperm. And these changes in membrane potential may help in the migration of proteins that are important in the sperm-egg fusion to the sperm head. And one such protein that we will be looking at as we move along is isomo. Now, capacitation results in the sperm to swim at higher velocities and generate greater force. And hence the sperm become hyperactive. So following capacitation, the sperms are able to swim better and are able to swim at a greater force, and hence they're called as hyperactive. The capacitated sperm also are able to show thermotaxis, which means that they're able to sense temperature differences and they swim towards warmer temperatures. The ampulla is considered to be 2 degrees Celsius warmer than the oviduct, and thus the sperm are able to sense this temperature difference and move and swim towards the ampulla. The capacitated sperm also show chemotaxis. Now, the oocyte and the cumulus cells secrete chemical attractants that are sensed by these capacitated sperm, and they're able to swim towards these chemical attractants, and hence they show chemotaxis. Now, progesterone, which activates calcium channels in the sperm's tail, promotes hyperactivity, and thus the presence of progesterone helps in sperm movement. Now, for chemotaxis to occur, it also requires the intracellular sperm to raise its pH. And thus, there's a lot of activity in terms of the sperm in the vicinity of the oocyte. Once the sperm reaches the ampulla and near the oocyte, the acrosome reaction can occur. This is a calcium-dependent exocytic process which results in the release of the acrosomal contents to the environment. We had seen these processes when we were learning about fertilization in sea urchins. Now, the principle is the same, where here we have in the blue box a sperm that is showing its acrosomal process, or the acrosomal vesicle that is shown in red. During the acrosome reaction, the acrosomal contents are released, and this is what is being shown in this figure. Now, once all the contents are released, then we can actually have the sperm's plasma membrane being exposed. However, there is a portion of the acrosomal membrane that is still present, and that region is called as the equatorial region, which is where the acrosomal membrane is covering the sperm cell membrane. The acrosome reaction usually occurs while the sperm are in contact with the cumulus cells, and thus the sperm have not bound to the oocyte in the case of mammals when the acrosome reaction occurs. This was shown in the figure that is shown in the yellow box. Now here we actually see 2 sperm, one in a white triangle, and the other one doesn't have any indication on it, but it is towards the right. When we look at the next box, and here in this case we can see colors, we can see that the green color to which the yellow arrow is pointing is the sperm that still has its acrosome intact. Now these sperms, both of them, one with the acrosome and the other to the left that does not have the acrosome, are interacting with the oocyte. However, the one with the acrosome is unable to fuse with the egg. And thus the acrosome reaction must occur before the sperm makes contact with the oocyte. Once the sperm is able to reach the oocyte, it interacts with the major glycoproteins that are present in the zona pellucida. Now, in the case of human eggs, there are actually 4 different major glycoproteins that are present in the zona pellucida, which are ZP1, ZP2, ZP3, and ZP4. Now, the sperm can bind to the zona pellucida protein ZP2 in humans. If we look at the figure that is shown to the right, if we look at the panel that shows ZP2, we can actually see sperm present there, which are those thread-like structures surrounding that circular oocyte that is there. And so, however, we don't see those thread-like structures, which are the sperm, in any of the other oocytes, which could have either ZP1, ZP3, or ZP4. Now, in the case of mice, ZP3 binding can trigger the acrosomal reaction. However, what you should remember is that in the case of humans, the sperm will bind to the ZP2 protein that is present in the zona pellucida. In the case of mammals, the side of the sperm head binds to the egg and microvilli to promote fusion. So the egg will have these microvillous finger-like structures, which we will see in the next slide, and the sperm interact with these structures for fertilization to occur. Now, the equatorial region of the sperm fuses with the egg cell membrane. And the protein usumo that is found in the equatorial region of sperm promotes the fusion of the sperm with the egg cell membrane. So let's just back up a bit and look at the figure that is to the right. And here we're seeing the sperm where it has its intact acrosome. We're looking at the equatorial region of the sperm. And there are these red dots, which are the usumoprotein. So the usumoprotein right now is present on the acrosome, and it is present on the plasma membrane of that acrosome, or in the membrane of that acrosome. Once that acrosome reaction occurs and all the contents have been released into the environment, we see that the usumoprotein is actually still remaining on the sperm head. And what we can see is it is found in the equatorial region of that sperm. Now, usumo is present on the membrane of the acrosome vesicle and is redistributed to the equatorial region during that acrosomal reaction, and this is an important step. The usumo will then react with juno that is present in the egg, and this recruits the protein CD9 to promote fusion of the sperm and the egg cell membranes. So if we look at the picture and go step by step, here is the first picture where the sperm that has already undergone the acrosome reaction has the usumoprotein on its equatorial regions, and it is going to interact with the microvilli that are present in the egg, and in this microvilli we have the protein juno. Now the sperm will actually interact with the egg on the side of its head. So it doesn't go perpendicular to the egg, but the side of the head of the sperm will interact. And so this way we can see that an equatorial region is actually interacting with the egg plasma membrane. We then will have the fusion where the sperm will be able to fuse its membranes with the egg cell membranes and thus enter into the egg. This is normally mediated between interactions with the usumo and the juno protein, and also CD9 will get recruited, and now we will have the sperm fusing with the egg cell membranes.
With this, we come to the end of our talk, where we learned about the different types of fertilization and different processes that occur in internal fertilization in the context of mammals.